Hi, everyone. I'm Dr. Regina McClinton, and it is my pleasure to welcome you to our initial episode of Health, Race, in Healthcare. And if you give me just a second, I want to do a couple things here. Still learning how to work Zoom. So thank you all so much for joining us. So glad to have you here, and really thrilled to have Dr. Amit Pai as our inaugural speaker. And so the goal of this Lunch and Learn is to share with people information about how race has been used or misused in healthcare, and to clarify information where race has been used and the wrong information is being utilized in the care of patients. And one of those areas is in understanding kidney filtration rate, and that is called eGFR. And so we are thrilled to have one of our own experts in that area, Dr. Amit Pai, to share with us today. So Dr. Pai, welcome.
Thank you very much, Regina, for the opportunity to discuss this topic.
Great. Yeah. So let's just begin and make sure everybody understands. If you could give us the definition, what is eGFR?
Sure. So the terminology eGFR stands for estimated glomerular filtration rate. And so, as you can imagine, it's a mouthful. So we prefer to call it eGFR. And this is a number that's basically representative of what we think of as kidney function. Now, the kidney as an organ has many functions. But for pharmacists, obviously, we think of it as main function as being that of removing drugs, which is, obviously not the entire piece of that function. But basically, when we're thinking about kidney function, the kidneys are responsible in a way of helping eliminate drugs. And that process happens through filtration, but also happens through a process known as tubular secretion, which is it's actually actively pumping out some of this drug. And for most molecules that we think about, they are able to just basically freely filter through to the kidney, through the nephron, through a capillary structure that's known as glomerulus that allows these compounds to freely flow through. And in most instances, those compounds will be reabsorbed into the bloodstream. So eGFR, again, is an estimation of that process because measurement is complex. So in practice, what would be required is we would have to administer some sort of substrate. Historically, that has been something called inulin, or iothalamate, or ihexol. There's a bunch of different chemicals that you can imagine that have to be administered in a known amount, and then we have to measure blood concentrations, or urine concentrations of those molecules, and that allows us to measure that process or glomerular filtration rate. Now, again, that is a complex thing to do in practice, and because that's complex, we have to estimate. And that estimation happens by basically measuring something that an individual that would produce endogenously. Mm-hmm. And so one of those biomarkers that we use is a biomarker known as creatinine. Okay. So I can talk more about that, or if you have questions or follow-up questions, I don't want to just keep going on and on.
Great. And if people do have questions, you can pop them in the chat and we will open up for questions at the end. But I want to talk about the fact that for African Americans, there are two measures of eGFR, and that is not the case for any other ethnic group. So let's maybe start that part of the conversation. Why are there two measures for African Americans in this country?
So when you say like two measures, you're referring to this idea that there's a correction factor in that equation, right? Right. So, taking a step back, our process for estimating glomerular filtration rate, at least over the last 2 decades, has relied on an equation known as the MDRD equation, which is known as the modification of diet in renal disease. So if you can think about that for a second, MDRD was the modification of diet in renal disease. And this idea was that if you reduce the amount of protein intake, you would basically help reduce the progression of chronic kidney disease.
Okay.
Now, that study was designed to basically test that. It wasn't designed to come up with an equation, but that study failed. So in that failure, what was recognized is that they had a great data set of measured glomerular filtration rate.
Okay.
And so one of the outcomes of that was to come up with an equation. When they came up with that equation, what they observed is that African American patients, and this kind of affects the terminology if you can think of it. When the MDRD equation came out, the actual term that was published was African American.
Okay.
And so I'll talk about the second equation in a minute, where we use a different term is used, right? So in that study, what they observed is, on average, patients that were African American have higher serum creatinine values.
Okay.
Now, as I mentioned, that's the endogenous biomarker that's used to estimate glomerular filtration. So to correct for those higher values, you have a higher factor. So it was only in that population that it was determined, but that was based on 197 patients out of a total of 1,000.
Okay.
Now, over time, that has evolved.
Mm-hmm.
In different ethnic groups, there's recognition that there's some other ethnic groups that have lower serum creatinine values on average.
Okay.
So for example, there's an equation for the Chinese.
Okay.
There's an equation in Japanese patients that have different correction factors. Now, again, the question of like why we need this, that I don't think was thought of in enough depth, to make sure that would make sense. And I think it's always been, I would say, an issue, like, whether that was reasonable or unreasonable to have put a race factor with the original MDRD equation. But the purpose was to basically, in that data set, as you can imagine, when an equation is created, it's created so that it's internally valid, meaning it makes sense with the information that you have.
Right.
Whether or not that equation is actually going to work for, you're studying 1,000 people, and in that 1,000 people, you have 197.
Right.
Whether that's going to actually work for the thousands of patients in real life is always an uncertainty. From that equation was designed for patients with chronic kidney disease.
Okay.
So that means if you have a serum creatinine that was above a certain threshold, or if you have an estimated. So when you think about this term, again, just taking a step back, eGFR is the estimated glomerular filtration rate.
Right.
And that exists across a spectrum, as you can imagine. So as it gets to as it gets below a certain amount, you're now at risk for requiring renal replacement therapies or transplantation.
Okay.
But from a drug dosing perspective, we care about the entire spectrum. We care about patients that are on the low end.
Okay.
And we also care about patients that are on the normal range.
Right.
And in some therapeutic areas, we also care about people that have higher than normal range values. So that equation, the MDRD equation, does not work for people across the entire spectrum. It only works for people with eGFR values less than what we would say is less than 60 ml per minute.
Okay.
Per body surface area normalized value, which is 1.73 meters squared. So because of that limitation, another equation was set up, and it's known as the CKD EPI equation.
Okay.
From the same group of investigators, and it included a larger spectrum of glomerular filtration. That equation also has a correction factor.
Okay.
But when that equation came out, it came out as a correction factor for Black patients.
Okay.
And I think that also, was recognized like, is this going to now be applicable
Right.
on a global scale,
Right.
not just in America, right? Because again,
Right.
when we're doing drug dosing, we're not just dosing Americans, we're dosing everyone.
Right.
And the US product labels that come out impact other countries. And so countries that have multi-ethnic societies,
Mhm.
you think about South Africa, you think about other countries within Africa,
Canada.
it
Right.
was
Yeah.
it was recognized that that equation didn't work.
Right. And I'm starting to laugh because, okay, so race is this social construct based for those of African descent on physical characteristics that really have no meaning: texture of hair, width of the nose, coloration of the skin. And yet it then becomes this indicator that your kidney function is different. And yet, people that were brought here as part of the slave trade came from multiple areas within Africa. So these are these, individualized populations, if you will, across the African continent that get lumped into one big sum, if you will. And then because of these superficial characteristics, you get told, guess what? Your kidneys are different. And so I'm so glad to know that the larger, more global version of this study said that estimation rate, this correction factor, doesn't really work.
Right. And I think that's always what's going to happen with any equation. Like,
Mm-hmm.
whenever you come out with an equation, whether it's going to work for an entire population is always going to be suspect, right? So an equation always is giving you an estimate of an average or central tendency value. So I think that's always something that people need to keep in mind. The challenge for us as pharmacists has
Mm-hmm.
been, just this focus on drug dosing, uh, basically relying on the number as an absolute value. When
Right.
in fact there's a lot of imprecision in that estimate, and trying to relaying that imprecision is a hard thing to do
Right.
because people want to react to a number
Yes.
in some way, right? So I think
Right.
that trying to message that in a way that's going to work has been a challenge
Right.
from the beginning.
Right.
And there's just there's too many equations. And in fact, from a drug dosing perspective,
Right.
these equations that we've just described are actually now just coming into use. So in the pharmacy literature, you have required something known as creatinine clearance
Okay.
instead. So I mentioned creatinine again as the biomarker,
Right.
right? So that biomarker, we can get a clearance term from that biomarker that
Mm-hmm.
relies on just age, sex, and weight of the individual.
Okay.
Does not include race,
Mm-hmm.
uh, but it also has its flaws as well. So, it's like, I think we can just keep going into this should be all about the flaws, just so many flaws. But
Right.
uh, it's, it's, um, it has presented a challenge now because, we're talking about this correction for race.
Mm-hmm.
But what's also happened is that now we're recognizing that we also need to, when we report these values, perhaps, I mentioned that these values are reported per body surface area,
Mm-hmm. Mm-hmm.
right? And the FDA issued guidance last year to change that
Okay.
to now not be body surface area based, but just simply be on a ml per minute basis. So what
Okay.
does that mean? What that means is that different ethnic groups are going to have, you know, uh, a better representation in a sense,
Mm-hmm.
because
Mm-hmm.
historically, you can imagine if you're dealing with developing a drug for a Thai population
Yes.
versus a Japanese population versus a South African population versus normalizing everyone to 1.73 meters squared as well is also wrong.
Right. Right. Right.
So
So it's the crapshoot.
it really is, because it's like, how do you convey that when you look at a
Right.
number, right? Because
Right.
it's just, you're not going to get all those nuances. Um, But hopefully that's what, pharmacy and going to pharmacy school teaches you, will hopefully
Yeah.
teach you all of those kinds of nuances.
Yes. So let's kind of make a little bit more of a connection with the practice of healthcare. Pharmacists are learning this information about these different um, algorithms that are coming out and what is best to use and how to use it. Um, I recently had my physical, so I get, my lab results, and it had eGFR, and then on a second line, eGFR black. And so my question is, what are doctors understanding and how are they responding to things? And are pharmacists being more informed, maybe more flexible on the topic or evolved, even, if you will?
I mean, I think it's a great question. I'm not sure I can completely answer that question on what the, how pharmacists take that information and kind of relay it to a patient necessarily. Part of what you described has been the standard for a few years,
Right,
primarily because, race information may or may not be in the medical record, as you can imagine.
right,
So it's been sort of something that the physician is supposed to inform the patient about, uh, without really thinking about what that means,
right,
right? So from a pharmacist perspective, most pharmacists, as you can imagine from the community setting, are never going to even see that number.
right,
There's been a lot of pushback on that. So years earlier, there have been initiatives on the national level to consider when prescriptions are made for drugs that require dose adjustment for renal
yes,
function, for kidney function. I said renal. I need to correct myself. Uh, Bruce is waving his hand correctly, which is, we want to be more patient centered. And so the correct word is to always refer to this as kidney function.
okay,
So anyway, uh, having that number on a prescription as an example, so that the pharmacist would have this information before
okay,
dispensing a medication, would
yes,
seem like a reasonable idea.
yes,
But there's been enormous national, disagreement on whether that's a good thing or not a good thing. So just to imagine just having a simple thing like that. The patient is informed
right,
with that number,
right,
but the pharmacist actually will not, most often in the community setting, have access to that information.
wow,
So it's unclear to me how a pharmacist would respond,
right,
without having to take additional time to find that information.
right,
So I
right,
think most of this, if it's happening, is happening in the inpatient side as opposed to the outpatient side.
wow,
But even in that, from that, in my experience as a clinical pharmacist when I was practicing, I have to just say I never was in that place where I would go and talk to a patient about their kidney
okay,
function and how I was modifying their dose. Like that interaction never happened.
right, right, right.
Um,
Interesting. So it seems as if there is this standard. Well, let me back up and ask this question. Is there any real benefit for measurements of GFR to have a connection to one's ethnicity or to have, a different algorithm or correction factor based on ethnicity?
so my personal bias on this, because again, we don't have any publications to kind of confirm this entirely, is that all of this stems from that biomarker.
Mm-hmm.
I think the fundamental problem is that serum creatinine is a poor, poor, poor biomarker of kidney function. There are just too many variables that can impact that value,
Mm-hmm.
from diet, muscle mass, as we've talked about,
Right.
and other factors. And some of these things are, what we're doing is we're relying on other variables to
Mm-hmm.
get us an estimate of what that production capacity is of creatinine.
Right.
So that's why we rely on age and and sex and these things, and there's been suggestions that race might have a factor.
Okay.
But the reality is, it's really hard to tease out.
Right.
It's really, really hard to tease out. And even, for example, because diet can have an influence as well.
Right.
Right. So if we were to move away from creatinine
Mm-hmm.
to something else, and that something else is something that has been, we'll say the new kid on the block,
Yeah.
which is 30 years old, but that's new in medicine,
Right.
and that would be cystatin C.
Okay.
And so if you rely on something like cystatin C, and you rely on equations that have cystatin C in them, they magically don't have a race factor. So you say to yourself, well, if I was using cystatin C and there's no race factor,
Right.
then why is there a race factor at all?
Right.
Right? Or if you say, well, if I take serum creatinine and I were to use it to estimate kidney function in children,
Yes.
right? And I say, I'm going to take equations that exist for children. Is
Right.
there a race factor? And the answer is no. So I think it's those kind of mathematical inconsistencies
Yes.
that
Yes.
kind of makes me think
Mm-hmm.
that, yeah, there's really not enough basis to
Mm-hmm.
kind of uh, have that role for race.
Right.
So
Wow. So now we know that measuring that factor is more a better understanding of what the kidney is and is not doing. It's been here for 30 years and recognized as an effective marker.
right.
Why are we still doing creatinine?
Right. So that's what I always get asked, like, why are we still doing creatinine? Well, because change is so slow, right? In
Yes.
medicine, change is so slow in medicine, and we're so used to tradition. And the bigger reason is just cost.
Okay.
So creatinine is, it's going to be pennies. Cystatin C might be $1.
Okay.
And that difference is huge from the perspective of, people are getting thousands and thousands of measurements
Right.
over their life. But another major reason is international standardization.
Okay.
So as you can imagine, with any lab value that's measured, you want to make sure that that value would be the same or similar if you had it measured in Detroit or if you had
Right.
it measured in Chicago or anywhere, right?
Right.
But there's been enormous variability on some of those kinds of measurements, and creatinine had a lot of variability associated with it.
Mm-hmm.
About 20 years, 25 years ago, again, I'm losing track of time here, like when it was, but at some point in time, a while ago, there was a move to do something known as IDMS, which stands for,
Okay.
again, a lot of acronyms here, but isotopic dilution mass spectrometry standardized serum creatinine.
Okay.
And so there was an international effort to do this. That has come to complete fruition now, so that, if you were to measure creatinine globally now,
Right.
in most instances, you're going to get a very similar value.
Okay.
Uh, and because of that international effort, now if you're doing a clinical trial in X country, right,
Right.
you have comparable measurements.
Okay.
That does not exist for cystatin C.
Okay.
So cystatin C standardization started about 4 or 5 years ago.
Okay.
So the National Institute's NIST, I forget what this National Institute of Standards and Technology, has just issued some guidance about 2 years ago or 3 years ago. And so that's kind of underway now, but it's
Mm-hmm.
going to have to follow the same kind of path as creatinine
Right.
did. So it's going to be at least a decade or more
Wow.
before we get there and before we can actually have a panel. So for example, we don't have that right now at Michigan
Right.
Medicine. We don't have cystatin C. It has to be ordered and has to be measured elsewhere, as an example.
Wow.
So if you get it, it has to be sent out to a reference laboratory, which is Mayo Clinic right now.
Wow.
Um, but we've looked at this because we've had some patients that have had those measurements, and indeed,
Mm-hmm.
for some drugs that we measure, like vancomycin. We
Hmm.
just actually this morning just finished up an analysis that shows that cystatin C would be better actually.
Okay. Okay.
So there's value. It's
Right.
just a question of the economics.
Right. Which is what a lot of healthcare and healthcare treatment comes down to. So if I'm a patient, right, and I have a bit more of a clue than most folks, I'm right, like the person my family calls when they have these kinds of questions,
Yeah.
and I'm having kidney function issues, and I say I want to have my CAT. I'm going to get that word wrong. CAT,
Cystatin C.
cystatin C.
Yeah.
If I want to have my cystatin C measured, what is the likelihood my doctor would even do that?
Sure. So what current guidance is from the National Kidney Foundation is basically when you have patients on, so I mentioned, this MDRD equation, it's good for individuals with eGFR values less than 60 ml per minute.
Right.
Again, so no, nuance number 9. so, so basically, when patients have eGFR values between 45 and 59,
Okay.
it is referred to as the creatinine blind zone.
Okay.
So in that range of values, you can get a little more imprecision in the estimate with creatinine because I mentioned there's processes of getting rid of creatinine. One is through filtration and the other one is through tubular secretion.
Mm-hmm, mm-hmm.
So you can have what's known as compensatory tubular secretion in that range.
Okay.
So you might accidentally not capture people correctly in that range. So
Right.
it's in that group, and also if they have certain amount of protein in their urine, they they should get the recommendation is to get a cystatin C measurement.
Okay.
So I would assume that if individuals were in that range and whatever, then they would qualify for getting reimbursed for that assay.
Right. Right.
Uh, but having said that, I think that's always going to be a conversation with the physician, to say I
Right.
just don't trust this, and I would like to have a more accurate estimate, um, and have that cystatin C measured then. There's, as you can imagine, multiple technologies on the horizon, even more expensive technologies that include like injecting individuals with fluorescent probes
Yes.
that exist. So the technology is out there.
Right.
It's just like getting to the point of like when can we actually have something simple. Simple always wins in medicine is the
Right.
challenge,
Right.
right? So that's why we are where we are.
Right. So we have a really great question that came
Sure.
through the chat that I want to share. And it says, as you mentioned, that vancomycin dosing was better with cystatin C, how would we adjust doing recommendations for medications if in the clinical trials for these meds, the measurements for eGFR were made with SCRC, and how would drug dosing adapt to the clinical level?
So that's a great question. I think from a modification of the from the request for modification of the actual product label for any individual drug, it's going to be very difficult for any drug company to make that kind of investment and actually change their label to help individuals modify their dosing. So what ends up having to happen is relying on publications coming out and for individual guidance changing. So thinking about Michigan Medicine, for example, I see Greg Eschenauer's on the call today. Like his group, as an example, antimicrobial stewardship team would look at the available evidence to see if they can change institutional guidelines. So Michigan Medicine, for example, made that decision um, a few years back, I think now, to say that different drugs are being approved on different equations right now. So
Mhm.
as I mentioned, there's MDRD, there's CKD
Right.
epi, there's Cockcroft-Gault, and now there's FDA guidance that says mil per minute and mil per minute 1.73 meters squared is going away. So, based on my math, there's going to be 9 potential versions of these equations in the product label, which is going to be a nightmare for pharmacists, right?
Yeah.
So, thinking about the antimicrobial stewardship group as one example, they made a decision to go with CKD epi as an example. And I think that's what the challenge is going to be, is that there's going to be institutional level decision to say, this is what we're going to do, with the recognition that on an individual patient level, maybe the pharmacist or physician is going to have to make an adjustment.
Right. Right.
But to answer the question that was asked, there's an opportunity to do that, but it just takes time, and it's a drug by drug thing. So with vancomycin, the likelihood of that changing is low because guidelines were just issued, which Dr. Mueller and myself are on as an example. We know how long it takes to get those out. That it's going to be at least another decade before
Wow.
we see another set of. So I think practitioners in the trenches are going to move on these things a lot sooner than
The
national
whole
guidance.
field. Yeah.
Yeah.
Wow. Wow. I think for lay people like myself, it is easy to forget how the pace of change in medicine, right? And you watch these shows, oh, I just read about this experimental thing. Let's just do it on this patient. And you think, wow, this stuff is really, and it's not that way.
It really isn't. I mean, it it follows that classic curve, right? So you have
Yeah,
any innovation is going to take whatever it is, 10 to 14 years typically,
right.
which is, I think would love to see the science and how to speed that up.
Yes.
Uh,
Maybe that should be our next faculty hire.
yeah, how to work on the laggards.
Right.
Yeah,
So I have one more question. Well,
sure.
two, and then we'll open up the floor for additional questions from the audience. And I'll give you both questions now. If um, you were an African American patient having issues with your kidney function, what would you want to know? What would you need to know to help advocate for yourself in regards to your doctor truly understanding how your kidneys are functioning? Because one of the things in the articles that we shared is that using the modified eGFR for African Americans, the issues of kidney function are not being detected early enough. So they're being told, oh, your kidneys are fine, but really they're starting to have issues that, if caught earlier, could be treated better. So I'll start with that question first. If you're an African American patient, what would you want to know, and what would you need to know to advocate for yourself?
I think what you need to know is, like, you have to be, obviously, if there's things that are impacting, I mean, part of it is going to be family history as well. So,
Okay.
is thinking about your family and thinking about, so again, you're putting me in this role, like I'm the patient, right? So I
Right,
would be
right.
thinking about my family and seeing sort of, what's happened to my family over time to see if there's that history, uh, and, and, with all of these measurements, again, there's going to be error associated with it. It's
Mhm.
really the trends, right?
Okay.
So, I would want to know from my physician, obviously, what sort of lifestyle changes I need to make
Hmm.
to help reduce the probability that I end up um, on hemodialysis, which would
Right,
be, really life altering and not something I'd want to end up on,
right.
as an example, right? So I really want to see how long can I avoid this, and I think, having listened to some of the nephrologists in
Mhm.
this space, there's really an opportunity, even when patients have poor kidney function,
Right.
to stay off hemodialysis for a long period of time, to
Okay.
really make lifestyle changes to ensure that you're not on that. So those are the kinds of things I'd want to hear. I'd want to hear, sort of like what's been done, who can I talk to, who are some patient support that I can connect with to have a buddy or someone to kind of make sure that I'm doing all the right things. In terms
Right.
of measurement, obviously, ideally, if my insurance or physician were willing to do this, I would love to have some sort of actual measurement of my kidney function
Hmm.
as opposed to having it be estimated,
Yes.
so that I know where I am, and whether it be, it's one of those things. Again, maybe I can't have this measurement every year,
Right.
but if I could have this measurement every 3 years or every 5 years, it's
Right.
still better than
Not
having
at
this
all.
uncertainty.
Right. Right. Right.
So, I would try to push for that and see if that would be paid for, um, or if I had the resources, I'd pay for it myself.
Right. Right. And last question before we open it up. Are there things that pharmacists can do to advocate for patients? And you talked about, if that number is on the prescription, that in the community setting supports the pharmacist in being able to advocate for the patient around medications and whatnot. What are other things that pharmacists maybe can or should do to advocate for patients in this area?
Yeah, I think it's definitely a perplexing problem, right? So because when we're thinking about this equation again, having this value becomes having this correction factor becomes an issue when you have patients that are on the edge of a dosing regimen
Mhm.
schedule, right?
Right,
So
right,
some drugs you may not be able to prescribe anymore if a patient's below that range. So if that race factor was taken out as an example, right? All of a sudden you may not be able to prescribe certain medications.
right.
So, if you were now measured out, if the estimate comes out as 28, for example, and without it, you're at 31 or 32. So I think what it falls on the pharmacist is to really think about, again, risk benefit,
Okay.
meaning that, how confident are we with that estimate?
Mm-hmm.
Should we make that decision to use or not use that medication based simply on that value? Is there something else we could measure to get confidence, even though the product label is going to have hard recommendations,
Right.
right? But it's really having that decision with the physician to say, we're not going to abide by this because it's in the interest of the patient to do this or not do this. So how do you actually do that? Like, how do you actually do it? It's is not straightforward,
Right.
to be honest with you. It's going to take some experience. So pharmacists that are doing this currently in the outpatient setting, or in the ambulatory care setting,
Mm-hmm, mm-hmm.
kind of hopefully they are going to start seeing more case examples of this and hopefully can publish in this space to say, we didn't follow this guidance. And if we didn't follow this guidance, the outcomes were not bad.
Right.
I think that's the kind of work we need to kind of really move this forward,
Right.
because that's not being captured right now.
Awesome. Well, this has been great. I want to now open the floor up for questions from the audience. And what you can do is unmute yourself and ask your question. So if we have questions from the audience, then please go ahead and do so. And Jasmine, please go ahead.
Okay. I was going to wait if anybody else had questions first, but that's okay. So I know we focused on how this can affect drug dosing, but are you aware of how this race correction doesn't have an effect on eligibility for kidney transplant? Like, is this leading to disparities in Black patients not being as eligible for kidney transplant compared to other races?
Yeah, I think that's a good question. Obviously, I'm not an expert in that space. That's why I kind of stuck with thinking about drug dosing, but I know there are data out there that suggest that this can impact. I think one of the studies that Regina shared with the group was kind of a survey of 50 some thousand patients, right, to look at to see how that would impact. And in that group, it didn't seem as clear to me that it had a major impact. I think part of the challenge again is that this correction factor is like having that correction factor of 1.16, for example, right? So it's the suggestion that it's 16% higher is really because the serum creatinine values are 16% higher. So we've looked at this at Michigan Medicine as an example. So I've pulled 45, 50,000 patients worth of data to look to see what is the average serum creatinine values. Does it kind of match up with, with these equations? And it does actually. So it's hard to completely answer the question, Jasmine, to be honest. Like I don't know. I know there's data that suggest that, but it's not clear to me yet.
Right, thank you. Any other questions? You can either unmute yourself or type it up in the chat.
I'm old and I remember when all these equations were started, but when I look at a patient and I calculate a creatinine clearance, let's say whether I'm using Cockcroft-Gault or I look at the eGFR or use any equation you want to use, I never believe that that's what the number is, right? So, and I don't think any seasoned clinician looks at a number, whatever comes out, 45 comes out, nobody really believes that it's actually 45 is the GFR or 45 is a creatinine clearance. Nobody believes that. In fact, when I would teach in therapeutics, I said the number is plus or minus 20% of that number. And honestly, tomorrow, it might be different than today. I don't know what blood pressure is. I don't know how my hydration status. I don't know what I had to eat. Creatinine, heck, any lab value itself is plus or minus 10%. And now you're going to put it into an equation. So it's a patient comes in, and I don't care what color they are, what ethnicity, race, whatever, they have in their body some GFR, and none of us can ever know what it is, and we don't know what it is. And we can use creatinine to make an estimate of what that is, or cystatin C, or something else, an exogenous marker. We don't know what it is, but we know it is that right there at that time. What math we use kind of puts us close to an estimate, but it's really only plus or minus 20%. Bigger dosing errors are made when clinicians treat that calculation as the gospel as what that is. A person really is at 45 without looking at clinical status. So I spent a lot of my years in the intensive care unit where a patient's blood flow to their kidneys changes drastically. It can change with any medication we give them. We give vasopressor agents, or patients become hypotensive, or patients are given massive fluid for fluid resuscitation. All those things have huge effects on blood flow through the kidney, and actually huge effects on serum creatinine. The measurement, actually, if you dilute them out with a lot of fluid, it's been shown you will make the serum creatinine number go lower, which will make you think you have really good renal function. You don't. You just have a fluid overloaded patient. The clinician never looks at that 45 number and says it's 45. The clinician looks at that number and goes, this patient is somewhere between, 35 and 55. And now let's look at their clinical status to see if, in fact, for example, if they're anuric, their GFR is 0. If someone's not making urine, the GFR is 0. The creatinine clearance is 0, no matter what the calculation is. And so it's that clinical expertise, it's that clinical judgment, it's looking at the patient, talking with the patient, that's going to make all the difference. And so the caution to the students is that number is not a real number. And going in knowing that it's imaginary, but it's an estimate, I think helps you with the dosing situation. Because honestly, in most cases, I'm in the middle of rewriting all the renal dosing recommendations for the UpToDate computer series. And most drugs, you're not making critical decisions with big dosing changes if someone is 39 or 42. Because really, they're somewhere like 35 or 50. You don't know what they are, and tomorrow they're going to be different anyway. So I am concerned about where I haven't got very excited about all these equations because I never really believe them anyway in my own clinical practice. But I do care about these questions like, are they eligible for a clinical trial? Are they eligible to get a transplant? Are we overlooking somebody because of some artificial construct that doesn't work for this person, and therefore we're not going to be aggressive with ACE inhibitors in this particular diabetic? That kind of thing is what worries me. Those are decisions that actually make a big difference. It isn't whether I'm giving cefazolin every 8 hours or every 6 hours. Honestly, that almost never is that important. So I don't know. I guess I don't even know why I'm talking other than to say stop believing that number. It's not an actual number anyway. It is only an estimate.
Yeah.
It'll only ever be an estimate.
I totally agree with what you're saying, Bruce. And I think a lot of people have had discussions on, like, again, how should we report these values, right? So Regina gave us the example of, like, she had this case and she got a value and it said this was the value, and if you're Black, this is the value. And so someone wants to interpret that value. And so if we now say, okay, let's make the move to saying it's 35, and then let's give a confidence interval or something, I'm just making that up, and say it's actually 29 to 43 or something, right? You can imagine, like, how would people respond to that? Like, how would physicians, and like how much education would have to go into explaining what that is now? And would that not be true for every measurement?
Right. And let me complicate my little case even more. I have my labs done at like 7:30 in the morning. That means no coffee. And because of COVID, it meant no water because I didn't want to go to a public restroom. So I've had no fluids this morning, and I go in and they take a blood draw and we're off and running. So really, we don't have an idea. And what if my numbers were really off and my doctor's all, oh, and so there are things that lay people aren't being informed about, like you should probably have a glass of water before you have a blood draw. That would be helpful. That we're not being told by our primary care physicians before we go into a lab to have lab work done. And then everything that you all have been saying. And I see Lydia has unmuted herself. So yeah. Lydia, please feel free to chime in here.
Well, I just think it's interesting because some of this discussion when we were talking about the data was a little bit unsatisfying, and like, well, how are we ever going to get to the right number? And then, I'm so glad kind of for Bruce's commentary, because it reminded me of exactly what we do in clinical practice. And it kind of puts the role of the pharmacist at the center of like, and it kind of reminded me, like right now we have this dosing thing that we have to do on inpatient, like click dots for renal dosing, which I think is silly, but I thankfully don't work inpatient. But it's actually the context is what we should be providing. And so I know when I'm asked the question about creatinine clearance and dosing, I'm always thinking in the way that Bruce kind of talked about it, because I remember in class, he gave us this example where everyone had to calculate creatinine clearance and every table had a different number. And then it was drastically different, right? Because if you use ideal body weight versus actual body weight versus blah, blah, blah. So I think that that's actually what maybe is more important, is to be thinking about the critical thinking that the pharmacist can provide, because we understand how these numbers are flawed, and helping dispel the dogma of if you're less than 50, you have to dose reduce the cefepime or something, which is silly because you don't. So I don't know.
I kind of want to turn around the words that Dr. McClinton had. She says, well, I go in and they have no idea what my renal function is. Actually, that's exactly what they have. They have an idea of what your renal function is, but they don't have an exact number. And it shouldn't even be our goal to get an exact number, because it doesn't matter, because it changes every day with diurnal variation and what you ate and all kinds of things, and your hydration status and all these kinds of things. You have an idea. And now dose off your idea. And should you adjust the dose of cefepime in this patient? I don't know. How sick are they? Where's the infection? How do I make sure I have a high enough concentration at that site of infection? Now, how aggressive do I want to be? That's a pharmacist's decision. That's clinical judgment. That's what's important. And you have an idea what that number is, but it is not hard law. There's art to this clinical practice, and there's science. Quit trying to think that this one number makes your decisions for you. You are the pharmacist. You make the decision based on looking at this patient, talking with this patient, looking at other labs, looking at cultures, all those kind of things. And you know what? Today, let's say the serum creatinine is 1.3, and Dr. Benita says, man, I'm going to be aggressive with this dose today. I'm going to hammer, this patient looks sick, they're septic as could be. I'm going to really hammer antibiotics. 2 days later, that patient may have the same serum creatinine. And she says, man, this patient's looking a lot better. I'm backing off on this dose. And that's great clinical decision making because it's not being driven by a number. And that's how I think one should practice.
Awesome. Dr. Hertz has unmuted himself, so we'll bring him into this conversation.
Yeah, I wanted Amit to get back to this idea of this sort of race-based fudge factor, whatever we're calling it. And, you know, we said it's artifactual because there's a lot of other variables that go into it. It could be genetics, it could be physiology, it could be behavior, it could be diet, it could be all these things that we can sort of get a surrogate of based on race right now, or as best we can do. So I've heard some discussions and some people advocating for getting rid of race in these kinds of formulas because it's not a real variable and doesn't have biological usefulness. I wonder, do you have any estimate of how much less precise our estimates would get if we just threw out race? And then looking forward, what do you think are the important actual variables that we would need to account for in order to get rid of race? What, when it comes to eGFR, what are the actual variables that you think race might be a surrogate of? And how much work would we have to do to replace race with the real variables?
So it's a lot of questions there, Dan. Okay, so I mean, my take on it again is that I think the reason we have all of these complexities and inclusion of race is because of creatinine. So if we go back to creatinine, this has been the rationale behind why these things might exist has been this idea of lean muscle mass being different across different ethnic groups. That's really never completely panned out as the only explanation. I think that we have to get away from serum creatinine. That's really what we have to go with. We have to kind of get to something like cystatin C or give something exogenously if we can. That would be ideal if we want an accurate estimate. The challenge is, how do you do that on a routine basis every single day for a patient? If you're monitoring them, that's not going to be feasible to do, right? So ultimately, it's just as been stated. I'm going to restate it again, but it's just an estimate. You know, how do we account for we were not going to be able to account for all of these possibilities in a simple enough way. So years ago, I worked with the FDA to try and sort this out. We came up with an algorithm with a lot of if and else type statements with 13 different variables that we should include. And it works. It improves the precision quite a bit. If you were to throw in BUN and albumin, and if you're throwing some other sort of measurements, the challenge is complexity, right? So now you have a 8 variable equation with 13. So you need. So again, could this be done? Yes, it could be done if this was all integrated within the electronic medical record. Again, it can be done by a system like Epic or whatever if they went. But then once you do that, how do you kind of rebrand or rethink about how to do this for all the drugs? And I think that's the challenge. Like, how do you modify labels? So it's just a very daunting task.
I had a question as well. So you said you wanted to get away from serum creatinine measurements, but I know that cystatin C is also secreted and reabsorbed in the tubules. So is cystatin C really a better measurement, or can we just get rid of race-based dosing, race-based correction factor in serum
Yeah,
creatinine.
actually, and so to kind of address a question that Dan had, thank you for that, Luciana, is that should we just throw that factor out, right? So I'm all about like the data's there. So the data that went into the generation of those equations exist. So what should happen is they should remove, if they were to remove that race variable, it would change the entire equation. So I don't think you should just take out the race factor without resetting all of the variables. So what I mean by this is you're going to get a different estimate for age. You're going to get a different estimate for whatever if you change that population level race factor. So that whole equation needs to be recalibrated. Now, the challenge of doing that, as you can imagine, is trying to educate everyone again about why you did that, right? But that would be the mathematically correct way to do it. And to your point about cystatin C, I completely agree. Again, no endogenous biomarker is going to meet all of the criteria that are necessary to give you an accurate estimate of the glomerular filtration rate. None of them are really going to pan out. And there's several others that I have not even talked about that have been tested that all have their issues. And so this is, again, a representation of this incremental change in medicine, right? So we just want to make a small change, and that small change will take X number of years.
There
A
we
lot
go.
of unsatisfying answers.
Well, I think it does, though, depict where the field is and how healthcare needs time to adjust, and the rate at which those changes happen. First of all, Dr. Pai, thank you so much. This has been fabulous. I personally learned a lot as a layperson. I'm sure all our pharmacists and PharmD students have learned even more, and really appreciate you being the guinea pig for round one here and participating. We will do this again next month, March 23rd, I believe, with Dr. Michael Smith, and we'll talk about racial bias in pain management. I want to thank all of you for attending. Really appreciate you giving us your time today. And we hope to see you again next month. Again, thank you, everyone. Bye-bye.
Thank you. Bye.
Bye.